Welcome to Bergeson and Campbell and the Actor Group's webinar, What's New with New Approach Methodologies? This is the second in our series of 2025 webinars. The entire list of our complimentary webinars can be found in the resources section of your portal there. My name is Lynn Bergeson. I am managing partner of Bergeson and Campbell and president of our consulting affiliate, the Actor Group. I am pleased today to serve as your moderator. We are so thrilled to be able to present this webinar to you today. We started planning for it late last year, and we're just delighted. We have a super distinguished panel of experts to talk to you today about the amazing developments in new approach methodologies. Because there are so many developments and there are so many things to discuss, we've extended our more typical 60-minute webinar format to 90 minutes to enable our distinguished presenters to give their remarks in an unhurried way and to accommodate all the questions that we have already received and that we anticipate receiving online during the course of the webinar. Now, before introducing our distinguished scientists, allow me to make a few administrative remarks. First off, the slide deck for today's webinar was sent to each of you who registered about a half hour ago. It is also available in the resources section of your portal. Your microphones are muted, but we really do encourage participation. And if you have questions or seek clarification on any remark that any of our presenter makes, please just put it in the Q&A and we'll take care of it at the end of the prepared remarks. And to the extent we are able to answer all questions, we always endeavor to provide answers in writing after the webinar. Full speaker biographies are added to the resource portal of your dashboard, along with some other, we think, very helpful materials that will help you navigate this complicated and exciting scientific space. So please check that out. And as noted, we're going to try to answer as many of the questions as we have already received and also get to questions that you provide during the course of the webinar. And what we don't get to today, we will get to. You don't want anybody to get grumpy that your question was unanswered because we really do try to answer every single question. So with that said, let me turn now to introducing our extraordinary scientists. We have three very distinguished scientists to present on this exciting topic. Leading us off is Adam Bettmann, a toxicologist specialist representing PETA Science Consortium International, EV. Adam, who is resident in the Twin Cities area in Minnesota, is going to provide a broad overview of currently available NAMs. Following Adam's remarks, Dr. Katie Paul Friedman will follow and give her comments. Katie will discuss the amazing work over the past few years of the New Chemicals Collaborative Research Program, the NCCRP. As you will learn, the body of work getting out of that group is both enormous and impressive. Katie is acting director of the Biomolecular and Computational Toxicology Division in the Center for Computational Toxicology and Exposure in EPA's Research Triangle Park Office of Research and Development. Finally, last but definitely not least, Rich, Bergeson and Campbell and the Actor Group's Director of Chemistry, Dr. Rich Engler, will give his presentation. Rich is going to give a broad overview of TSCA Section 4 testing authorities and integrate some of the concepts that we learned from Adam and Katie and talk about how new approach methodologies can serve OPPT submissions. So with regard to the agenda, we promised in our promotional materials and deliver here on three broad topics: NAMs and their use and submissions to OPPT, the New Chemicals Collaboration Research Program generally, and TSCA Section 4 Chemical and Testing Authorities. With that, it is my pleasure to turn the webinar over to Adam Bettmann. Okay, Adam.
Thank you, Lynn. Starting off today, I thank you for the opportunity to speak to you about new approach methodologies for TSCA in 2025 and beyond. Next slide. So for those of you who aren't familiar with my organization, we are made up of 23 scientists located in the US, UK, Germany, Switzerland, Belgium, and India. We focus on advancing reliable and relevant approaches for regulatory toxicity testing by funding and designing method development and validation efforts, organizing free training opportunities, workshops, and webinars, conducting retrospective data analysis, participating in various standards-making organizations and committees, and publishing and presenting on our work. In everything we do, we work collaboratively to ensure that we end up with robust testing approaches that can be used by industry and accepted by regulatory agencies. Next slide. So for our outline today, first I'll give a brief overview of what NAMs are and explain both how they are helpful in the field of toxicology in general and then why they are important specifically for TSCA. I'll then discuss opportunities to use well-established or guideline NAMs, as well as opportunities to develop and use fit-for-purpose NAMs when a guideline doesn't exist. And lastly, keeping up with the quickly expanding field of NAMs can sometimes seem overwhelming, so I will provide some training and educational resources. Next slide. So first off, what are NAMs? There are various definitions, but basically new approach methodologies are defined as any technology, methodology, approach, or combination that can provide information on chemical hazard and risk assessment and avoid the use of animals. These may include in silico, in chemico, in vitro, and ex vivo approaches. In general, NAMs provide many advantages in toxicology testing. The first is human relevance, meaning that they can use cells or tissues from the species of interest, usually humans, and using these types of test systems that are relevant to human physiology and anatomy can help accurately predict toxicological effects. As far as logistics go, many NAMs provide results rapidly, and using new approach screening assays can help justify either no need to continue with additional testing or inform the need for more additional and targeted testing. NAMs can also provide a deeper understanding of toxicological effects, as many in vitro methods can provide quantitative mechanistic information down to the cellular level. And the use of quantitative metrics to evaluate toxicological endpoints of interest can allow for better reproducibility than methods that rely on qualitative observations alone. Lastly, testing on animals continues to raise ethical concerns as millions of animals are bred specifically for toxicology testing. So if we can reduce and replace animal test methods, we can limit the impact on animals while optimizing human relevance, efficiency, and data quality. Next slide. So how are NAMs specifically relevant for TSCA? Well, due to the benefits outlined on the previous slide, NAMs are being incorporated into many global chemical regulations, including TSCA. And as many of you may know, in 2016, the Frank R. Lautenberg Chemical Safety for the 21st Century Act amended the original 1977 Toxic Substances Control Act. Specifically, Section 4H of this amendment called to reduce and replace, to the extent practicable and scientifically justified, the use of vertebrate animals in the testing of chemical substances or mixtures, and promote the development and timely incorporation of alternative test methods or strategies that do not require new vertebrate animal testing. This amendment also required the EPA to develop a strategic plan on this topic, and in 2018, the EPA published its strategic plan to promote the development and implementation of alternative methods within the TSCA program. This strategic plan has three core components that include identifying, developing, and integrating NAMs for TSCA decisions, building confidence that the NAMs are scientifically reliable and relevant for TSCA decisions, and implementing those reliable and relevant NAMs for TSCA decisions. In addition to this, in 2020, the EPA created a NAMs work plan that was a joint effort between the Office of Research and Development and the Office of Chemical Safety and Pollution Prevention. It addresses the reduction of animal testing through the use of NAMs while maintaining protections for human health and the environment. And this work plan encompasses a broader scope than just TSCA and was most recently updated in 2021. Next slide. So now that we have a broad overview of the importance of NAMs, both in the scientific field and specifically for TSCA, we can take a look at some of the currently established methods. When thinking about well-established test guidelines, the Organization for Economic Cooperation and Development, or OECD, might be the first organization that comes to mind. This organization maintains test guidelines that are recognized internationally as standards for chemical testing. And as you can see in the text here, works to ensure that testing keeps pace with scientific progress and that they promote the 3 R's of the replacement, reduction, and refinement of animal experimentation. This effort has led to the acceptance of many new approach test guidelines. Some of these guidelines can then be used in published defined approaches, or DAs, and integrated approaches to testing and assessment, or IATAs. These prescribe how to apply multiple test guidelines to answer a defined hazard characterization question and can potentially inform risk assessment by weighing all of the available evidence. In addition to test guidelines, DAs and IATAs, OECD also publishes case studies demonstrating how to apply specific approaches to broader scenarios, detailed review papers. Oh, I'm sorry, can we go one slide back still? Let's see. Detailed review papers that provide an in-depth analysis of a specific topic related to chemical safety and environmental health, and validation reports, which are comprehensive documents detailing the validation process of a test method. The link at the bottom of this screen takes you to a useful OECD webpage with multiple resources. The webpage also has a link to the work plan of the test guidelines, which lists the methods under development. This document is published annually and is a very good resource to keep up to date with what new test guidelines are expected in the coming years. All right, next slide, please. So shifting from international to national, the US EPA has its own guidance on testing. Examples here include the 2018 policy on the use of NAMs for skin sensitization testing, and more recently, a decision framework for the hazard identification of eye irritation and corrosion for new chemicals. This framework notes the EPA's preference for the use of NAMs in this context, and that it does not encourage the prospective use of the in vivo rabbit eye test. And similar to this, the EPA does have frameworks for skin irritation and skin sensitization in the works, which will hopefully be published in the coming year. I'll also point out that as required by the amended TSCA, EPA has published a list of NAMs that they have identified as scientifically reliable, relevant, and capable of providing as good or better information than obtained from animal testing. And while this is still a useful list of methods, it has not been updated since 2021. TSCA 4H2C does state in its requirement that the list shall be updated on a regular basis, so hopefully it will be updated in the future. Fortunately, though, the list includes text that specifies that it is not exhaustive and that the EPA will review any NAM it receives, and it also encourages all stakeholders to consult with the EPA on the development and or use of NAMs. Next slide, please. And speaking of consulting with the EPA on the development and or use of NAMs, more recently, the EPA published a report on statutory and regulatory requirements for vertebrate animal testing and flexibility for implementing NAMs. This is in July of 2024. And it reiterates that the statutes did not generally preclude the use of scientific information or data from NAMs as long as the contexts were well defined. The alternative method was fit for its intended purpose, and the NAM's advantages and limitations were transparently evaluated. Next slide, please. Now, there may not always be an accepted and standardized guideline available to assess your potential endpoint of interest, which is where the development of NAMs that provide fit-for-purpose solutions come into play. So in doing this, we first need to understand what goes into a successful NAM. These graphics provide the key concepts in establishing scientific confidence in NAMs, and they look very similar. One was the result of a collaborative publication between my organization, ECFAM, the OECD, NCP/NICEDM, and the US CPSC, and the US EPA that came out in 2022. The other is a collaboration between the then 17 US federal agencies that comprise ICFAM, which came out last year, March of 2024. You can see that they both include consideration of human biological relevance, technical characterization, fitness for purpose or context of use, data integrity and transparency, and independent review. All of these concepts should be top of mind when developing NAMs. Next slide. And when we say fit for purpose, what we mean is a method that is well equipped or well suited for its designed role or purpose. This graphic here is tailored specifically towards considerations that need to be made when developing a NAM in the inhalation toxicology space, and I will use it as an example. These considerations include the chemical properties of the substance or substances you wish to evaluate, what model system to use, what exposure type, exposure setting, exposure frequency, and exposure duration to consider, and then also which sector or regulatory agency or even international agency your NAM may apply to. So as you can see, there is an extensive thought process that must go into developing a NAM. Next slide, please. A good example of this process comes from a submission for the pesticide chlorothalonil as part of the Federal Insecticide, Fungicide, and Rodenticide Act, or FIFRA, and it nicely illustrates where a fit-for-purpose proof of concept can be developed, proposed to the EPA, and then eventually used in risk assessment. You can see from the timeline here that this was roughly a 5-year process that was initiated by the agrochemical company Syngenta in 2017. They developed a proof of concept new approach to test the inhalation toxicity of chlorothalonil. This was then presented to the FIFRA Scientific Advisory Panel in 2018, and the Office of Pesticide Programs, or OPP, continued working with Syngenta through 2019 to address the recommendations from the SAP. These efforts were successful, and in 2021, OPP published the draft risk assessment for chlorothalonil that included the first application of this new approach method. And then lastly, due in part to this successful application, the following year in 2022, the OECD published a case study on this new approach for potential future application. So in all of this, I'll emphasize that they followed the key concepts for NAM development that I mentioned earlier, and that the EPA were wonderful, helpful collaborators in the process. Next slide, please. And since there continues to be a great deal of work still needed in the inhalation toxicity space, we at PETA Science Consortium International are using these same principles to help build scientific confidence in the use of human cell-based methods to predict respiratory toxicity in humans. One of our major projects over the last several years is the In Vitro Systems to Predict Respiratory Toxicity, or INSPIRE project, where we have examined 2 classes of chemicals using different routes of exposure and 2 human cell-based biological test systems. A manuscript for our work with silanes has been published, referenced at the top of this slide, with another for our surfactants work in progress and will be coming soon. Our goal for 2025 is then to integrate these results into an OECD test guideline. Another slightly newer inhalation project that we've been working on is a collaboration with the EPA New Chemicals Division to help provide standardized frameworks for the evaluation of new chemical surfactants and polymers that may be hazardous via inhalation. These frameworks leverage NAMs to provide a transparent, efficient, and reliable evaluation of these chemical categories. And we do have 2 posters that will be presented at the upcoming Society of Toxicology conference this March in Orlando, referenced here at the bottom, if that is something that interests you. Next slide, please. So in closing, if you want to stay on top of the quickly developing field of NAMs and toxicology, we do have some great resources for you. The first is the EPIC webinar series, co-organized by the U.S. Environmental Protection Agency, our group, the PETA Science Consortium International, the Institute for In Vitro Sciences, and the California Department of Pesticide Regulation. We also have a whole host of past webinars that are available to you on the PSI website in our webinars section. The EPA has assembled a catalog of NAMS training materials on their website that includes multiple years of training presentations, slide decks, and helpful NAMS-related resources. The European Commission has a tracking system for alternative methods towards regulatory acceptance, or TSAR. This allows you to search both methods that have been accepted or in the acceptance process and provides really nice visuals as to where those methods stand. And then the NTP, Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICETM, also currently has a list of accepted methods on their website, and they are working on an exciting new project called the Collection of Alternative Methods for Application, or CAMERA project, which is expected to be a detailed and user-friendly, really one-stop shop for information about NAMs. So we're looking forward to that. And finally, if you are more of a visual and hands-on learner, the Institute for In Vitro Sciences does offer a week-long practical methods workshop 3 times per year at their headquarters in Gaithersburg, Maryland. And I have gone through that, and it is fantastic if that's something that interests you. This is a busy slide with many links, but for those resources that I mentioned on the previous slide, they are listed here. I'd encourage you to check those out, and then along with other references from items I mentioned in my presentation, if you'd like to dig any deeper on some of the topics that we discussed today. Next slide, please. And as always, if you have any questions that come up, please do not hesitate to contact me. For all of your new approach methodologies questions, even if I don't have the answer, I can likely put you in touch with either experts on our team or those that we collaborate with in order to help you out. I really thank you for your time. It was a pleasure to be here, and I will turn it over now to Katie Paul Friedman.
Well, Adam, thank you for your presentation. I really enjoyed it. I'm really happy to be with all of you here today to give you an overview on the progress of the New Chemicals Collaborative Research Program. And as I start, I'll just let you know that I work for the US EPA Office of Research and Development, and the views expressed in this presentation are my own and do not necessarily reflect the views or policies of the US EPA. So given the presentation we just saw from Adam on TSCA and the framework there to allow for the development and use of NAMs, I don't need to belabor the point that there's a key challenge that's being addressed by the New Chemicals Collaborative Research Program, or NCCRP, which is that many new chemicals have little to no chemical-specific information available when EPA is asked to review them. And currently, the program uses QSARs, read-across, other predictive approaches for data gap filling. But what we would like to do in the Office of Research and Development is to augment those currently available approaches and to develop and make publicly available new QSARs, new approaches that follow OECD development guidelines. And the reason that this has come to a fore, I think now, is that implementing those changes that Adam spoke about in 2016, the Lautenberg amendments to the Toxic Substances Control Act, really increased the resources that would be required to review and make determinations for all new chemicals, as well as documenting and supporting those determinations appropriately, and then further identifying reasonable conditions of use. And so our job in the Office of Research and Development is to create new approach methods, along with providing support on data curation and the development of decision support tools that will help address this challenge by providing analog approaches, groupings, addressing hazard data gaps, identifying potential conditions of use, and in general, just furnishing more information to make the required determinations efficient and reproducible. And in the Center for Computational Toxicology and Exposure, where I work, we are well situated to contribute to the development of NAMs for this purpose. You may be familiar with this 2019 paper on the left, the Next Generation Blueprint of Computational Toxicology at the US EPA, led by Rusty Thomas, our center director, and a number of us working in the center at that time. And basically what we put forward was here in the middle of the slide, the idea that all of our work is centered on 4 principles: chemical characterization, hazard evaluation, toxicokinetics and in vitro disposition methods, and then exposure assessment, and providing computational high-throughput approaches to do that. This is supported by our efforts to establish scientific confidence, which was also mentioned by Adam, to characterize uncertainty and variability, to perform outreach and training, and then to also develop software and decision support tools that are publicly deployed so that the public and other stakeholders can see the types of tools that make this NAM information available. And I have too many NAMs and approaches to list here on the slide, but just to give you an idea of the types of projects that we're working on in the center that cut across not only the NCCRP, but existing chemicals and other stakeholders beyond pesticides and toxics. So I'll give you an overview today of the 5 research areas that we've planned for the NCCRP. They're contained in this graphic to the right. First is approaches for chemical categories and analogs. Second is developing and expanding databases of information that's relevant to TSCA chemicals. Third is the development of quantitative structure-activity relationship models and other predictive models. 4 is in vitro assay data and models that we can put into a battery for estimating NAM-based points of departure. And then the final one that I'll talk about today are those decision support tools that we've been developing in collaboration with OPPT, New Chemicals Division. First, I want to show you that we haven't just started, although we are in the nascent stages of this program. We first started discussing the potential for this program in 2021. In 2022, we had a series of public meetings, including a review by our Office of Research and Development Board of Scientific Counselors, which is our federal advisory committee that advises our research and development. And in 2023, we really began the work. This was the beginning of our Strategic Research Action Plan, which guides all of our research in the Office of Research and Development. And at that time, we selected and sourced chemicals for proof of concept work with in vitro NAMs within the NCCRP. In this last year, we've really ramped up our engagements with the New Chemicals Division using a decision support tool called the Chem Informatics Modules, which I'll show you some screenshots of, as well as some other ORD tools for new chemicals. We've made progress on early publications on our chem informatic approaches, which I'll detail here today. And we've begun our in vitro NAM data collection for the chemicals we sourced in 2023. Moving into this year, we are working in earnest on the development of case studies using those ORD tools for new chemicals, finishing our data collection for a proof of concept with in vitro NAMs, and then continued development of QSARs. And then looking ahead to next year, we'll be thinking about trying to publish more of our in vitro NAM work and other tools work, and moving on to our next research action plan cycle. So we have to plan for our next ORD cycle next year. So Research Area 1 is the one that connects the current approaches in OPPT New Chemicals Division with where we would like to supplement category and grouping approaches. You may be familiar with the 2010 New Chemical Categories. The document is pictured here on the left with the link. And these are 56 existing new chemical categories that are largely characterized by shared structural features, in some cases by shared physicochemical properties. And these chemical categories are typically used to identify potential hazard concerns and testing strategies for new chemical submissions. So the goals of our research are to implement chemical categories in a transparent and reproducible way that's easy to update with new information, additional structure, feature descriptors, physical chemical data, or even NAM data. And then the other thing that we would like to do is make it so that you can computationally assign these 56 existing new chemical categories so that someone doesn't have to eyeball the structure. Rather, we can computationally assign a category based on a computational fingerprint of that structure. So to give you an update on some of our published research, I'll spend a lot more time on this publication, a chem informatics workflow to select representative TSCA chemicals for NAM screening as it relates to the in vitro screening that I'll describe today. But we have some other work ongoing as well. We've made progress on an automated approach to chemical grouping via application of the European Chemicals Agency assessment of regulatory needs categories. We've also made progress on benchmarking read across to enable application of ORD tools like generalized read across, known colloquially as GENRA. This is a computational way of doing read across that is reproducible, systematic, and uses neighborhoods of chemicals rather than a single target to analog relationship. And then finally, I'll talk a little bit about creating new categories using structure and obtaining greater coverage of the TSCA active inventory with this newer approach. So first, a little more detail on our collaboration with the European Chemicals Agency to try to computationally assign the categories that they have assigned to chemicals. There are currently 86 groupings on their website that contain over 2,000 substances. And one might ask the question, how can these categories be assigned computationally rather than by expert judgment and just looking at the structure? So what we've done is use structural Morgan fingerprints, it's that chemical structure fingerprint, to describe the chemicals that are found in those groups on their website. And then we built a machine learning model to predict the chemical groupings, and we use our EPA DSS Tox chemical library as our test set, where we have over 1 million chemical substances. And many of the substance groupings out of those 86 are similar or overlapping, and so these models provide programmatic support for groupings and which ones might be more likely than others to be appropriate for the chemical structure. And as we further develop ORD tools, we could implement this kind of model to allow for computational prediction and preliminary category assignment to the ECHA categories. So this could be done before a chemist even really looked at the structure, as long as we had the computational smiles that represent the structure. Next, I want to talk a little bit more about benchmarking generalized read across in this paper that we published led by Grace Padowich this past year. And what we did here was really take industrial chemicals and look at how well generalized read across, or GENRA, would work in that chemical space. So to do this, we again collaborated with the European Chemicals Agency, and they publicly release REACH submitted data dossiers through a program called iEuclid, and their version 6 is what we use to extract information on nearly 400 unique target source analog pairs. This led to 5,000 read across instances in that database. And the first thing we asked was, well, how similar are the target to analog pairs represented in that data set? So the graph to the left on the y-axis is the number of substance pairs target to analog. And on the x-axis is the Jacquard structure similarity, where 1 means that the pair were identical and 0 means no similarity at all. And you can see this bimodal distribution where there were a number of pairs where they're very similar, but then if you look at this other part of the distribution, perhaps the median structural similarity might be 0.4 or even less. And so that gives us some understanding that read across in practice in REACH dossiers has a range of structural similarity. We also wanted to evaluate how well GENRA would have done in predicting points of departure or effect level doses compared to what was demonstrated in the read across pairs in iEuclid. And we found that for 65% of the chemicals in these pairs, GENRA predicted a POD that was less than what was predicted using a single target to analog relationship from these REACH dossiers. To the right in this graph, you can see on the y-axis is the empirical cumulative distribution, where 1 means 100% of the chemicals in the data set and 0 means 0% of the chemicals in the data set. And to the left are larger PODs, and to the right are smaller PODs. And the GENRA predicted POD is in red, and the ECHA iEuclid PODs from single target to analog read across are in blue. And what I want you to notice is that while GENRA, for many of the substances, predicted a POD that was smaller, it was not smaller by much. The differences in predicted POD are pretty small when you consider the variance in replicate points of departure from replicated in vivo toxicity studies. And really the strength here you get from generalized read across is instead of having that single target analog relationship define a point of departure, we now can define a neighborhood of structures that may all be relevant to predicting the point of departure for that target. So the third paper I want to describe for you in a little more detail is that cheminformatics approach to grouping the TSCA non-confidential active inventory and then selecting the chemicals for our proof of concept in vitro NAM screening. This was published just last month. And to the left, you can see a generalized representation of our workflow to do this. We took the TSCA non-confidential active inventory. Only about half of those substances have a defined structure. Nearing 15,000 substances. And for those with a defined structure, we profile them through our computationally assigned new chemical categories, and then also something called classifier, which is a taxonomy. Most people think of taxonomy like biological taxonomy. Classifier is taxonomy for chemistry on the basis of structure. So you have hierarchical classification simply based on the structure features. And we basically use that to create primary structure categories, secondary structure categories, and then finally terminal clustered structural categories to then select about 300 representative substances by structural diversity. And to the right, I can explain this figure a little bit. You don't really need to see as much detail. You can kind of zoom out and understand what you need to know. So in this T-SNE plot, each dot is representing a chemical based on its structural features and their relationship to each other in space. If they're very close, they're very similar. And this is looking at the entire structure universe for the TSCA active inventory. And in orange are those chemicals that we've identified as being amenable to aqueous in vitro-based NAM screening. And then in the figure to the right, what we've done is show you in blue the entire landscape, and the orange dots are the chemicals that we selected for our in vitro NAM proof of concept screening. And what I want you to see is that those orange dots are really scattered throughout that whole blue cloud. So we've endeavored to use categories to essentially pick those representative chemicals so that we can generate NAM data on a much more diverse chemical space that's relevant to industrial chemicals. Research area 2 is about developing and expanding databases. And this is important because if you want to do something like generalize read across or build QSARs or make any kind of inference based on an analog, you need to have analog information. So this is our effort to try to increase the amount of data available for those types of inferences. We're also expanding our data interoperability by making it possible to consume iEuclid data, such as data from ECHA REACH dossiers or data made available by Health Canada, and to standardize ORD data into that format so that they can be interchangeable. And then we have plans to curate data from PDFs on ChemView, which is publicly available, into computationally accessible data. So rather than having the data be in the sort of image file of a PDF, we'd really like to extract the numbers and the pertinent information so that we can use it for modeling and rapid computational inference. Research area 3 builds on that concept of making more data available, then we can build more models. We're in the process of building a QSAR model for prediction of repeat dose point of departure values using the EPA ORD database, ToxValDB. We've also been building new quantitative structure use relationship models, which I'll talk a little bit more about in a coming slide. And then for exposure, we're developing a number of different exposure models, but the one I want to key up for you today is the development and publication of QSARs for predicting chemical occurrence in over 30 environmental or biological media. So first, the QSAR for repeat dose point of departure. This would be obviously very useful if you could immediately know a potential dose and then some confidence interval around that dose at which you'd expect that chemical to be active in repeat dose animal studies like a 90-day study or the non-cancer portion of a 2-year study. So to do this, we've used estimates of point of departure from traditional toxicity information that's curated into the public database, ToxValDB version 9.6.1. We take those estimates, we develop machine learning models, and those give us really reasonable error, especially given the variability in traditional toxicity information. That's represented in the graph to the right, where you can see predicted point of departure on the y-axis, and on the x-axis, experimental point of departure. And our error is represented as root mean square error, approaching about 0.7 log 10 mg per kg per day, which is actually pretty similar to the error just if you repeated a 90-day study multiple times. The other nice thing about this model is that you can directly explore the applicability domain of the model and look for well-represented or less well-represented chemical structure classes. So I've taken some of the most represented classes and show them in the figure to the left. Class here is coming from classifier. We use it again for chemical grouping. And you can see examples like triazoles, amino acids, halobenzenes, ethers, amines, carboxylic acid derivatives, etc. And then you would know exactly how many constituents of those classes were actually used in the training set for the model. And this will be made publicly available and also available by API. Next, a little bit more detail on those quantitative structure use or QSAR models and QSARs for media occurrence. I get really excited about the QSAR because I think this is just such an amazing idea that Kristen Isaacs and Catherine Phillips in our center had, where they basically take product data sheets from existing products, they curate out which chemicals are there for what purpose, and then they can annotate those structures as belonging to a particular functional use, such as for catalysts or crosslinkers or colorants. And then we can actually look at each of those functional uses and figure out how many of those chemicals are actually within domain for our quantitative structure use relationship models. Similarly, for the multimedia monitoring database, Kristen Isaacs has led a group where they curate information on the presence of different types of chemicals in different types of environmental media. That then allows us to understand the types of chemical structures that are present in different environmental media, so that for some new chemical structure, we can predict what kind of environmental media we might end up finding it in. This is really relevant to exposure because if we know which environmental media might contain that structure, we can make a better prediction of whether or not humans will be exposed and at what concentration. Finally, I want to talk a little bit about research area 4. This is our in vitro assay data and models research area. I mentioned we're working on a proof of concept here, and this proof of concept answers a number of research questions. So in this figure, I've looked at inference level versus outputs from in vitro NAMs. So you can imagine we can simply look at NAMs for demonstration and evaluation. Do they work? We can look at NAMs for informing specific chemicals for their assessments. We can look at NAMs for developing information for analogs. We can look at NAMs for developing information on known categories or groupings. And then we can also build QSARs on top of the NAMs so that instead of generating the NAM data, we could actually predict those outcomes. And different outputs from various in vitro NAMs cover different inference levels. Much of what we're doing covers all of these inference levels. We've been refining the chemical domain for screening, really trying to understand before you screen a chemical, is it appropriate to the assay you're putting it in? We are working to estimate point of departure using an in vitro NAM battery, and I have a few more slides on that. We are working to potentially inform data gathering or data gaps for specific hazards, such as developmental toxicity. And then I won't speak about this much today, but Adam gave you great insight into what's being done in inhalation, and we have some work ongoing here as well, looking at specific chemicals and how we can predict their inhalation risk. So moving on from here with a little bit more focus on our proof of concept design, what are the assays that we've included and how are we going about doing this? This proof of concept design was included in our 2022 report to the EPA Board of Scientific Counselors, and you can find it online. But essentially, we've published this first part, that cheminformatic approach to selecting representative chemicals to screen. That part has been completed. Now we're really focused on generating the data. And today I'm mostly going to talk to you about the human health component, but we have a number of other pieces here as well across ecological health and inhalation exposure. For ecological health, we're working on modified in vivo studies that are high throughput, as well as high-throughput transcriptomics in those models. This is pilot work with a small number of chemicals. We're also working on optimizing a system and identifying aerosols of interest for inhalation exposure. And what I'll focus on in the next couple of slides is what are we doing for human health in terms of combining broad and targeted screening assays. And this isn't coming sort of out of nowhere. We've been working on this for a while. You're probably familiar with this work throughout the field. I'll touch on just a couple examples. These two examples happen to come from work that I'm lucky enough to be involved in. The first figure on the right represents work that we did within the Accelerating the Pace of Chemical Risk Assessment, or APCRA consortium. If you're not familiar with APCRA, this is a group of scientists that work at regulatory agencies across the world who are essentially developing case studies of applying NAMs to various contexts of use. And so what we did in our first paper to the left in 2020 was we looked at data-rich chemicals and developed a NAM-based point of departure based on all of the in vitro potency data from ToxCast and a high-throughput phenotypic profiling platform contributed by A*STAR, which is an organization in Singapore. Then on the right is what we've done since. This was recently accepted at Toxicological Sciences. In this work, we look at 200 chemicals, including more data-poor industrial use chemicals, and we generated the data for these chemicals to be able to develop a NAM-based point of departure using a specified battery of broad and targeted assays, like you see in the yellow module of this process. We increased the complexity of the in vitro to in vivo extrapolation approach and a number of other analysis upgrades so that we can basically start to move towards what does a NAM-based assessment look like that creates a NAM-based point of departure and some other early indications of hazard. Other people are working on this too. Cosmetics Europe has explored similar toolboxes for a NAM-based point of departure for cosmetic ingredients. In this paper in 2021, led by Matthew Dent, there's a whole workflow that starts with use and exposure and proceeds to an in vitro NAM battery, which I've boxed in red. What you'll see is interesting. It's just like our NAM-based battery. We're pairing broad profiling NAMs like high-throughput transcriptomics, which I'll talk a little bit more about, and targeted NAMs like in vitro pharmacology profiling, which is what's used in the pharmaceutical industry to look at specific receptors, ion channels, other biochemical targets that are associated with off-target liability. A lot of this experience is built on the US EPA's ToxCast program, which actually is housed here in the center that I work in. And the targeted NAMs that tend to be included in these toolbox are customized to the potential use, exposure, indication, known mode of action. So that tends to fluctuate. And this really reflects early practice now of pharmaceuticals, been doing it for a very long time, but also cosmetics and personal care product sectors. So just some more examples in the literature to show that we're in very good company. This is on the left, an excerpt from a paper by Alistair Middleton and colleagues in 2022. And what I wanted to pull out was a figure that indicated they're using high-throughput transcriptomics, or HTTR, in 3 cell lines. They're using a cell stress panel, and they're using in vitro pharmacological profiling, so a panel of multiple protein-based targets. And Sophie Cable leads this progress work in 2024, same group of people looking at personal care products. And they have an in silico tier 0. And then interestingly, their tier 1 is a systemic safety toolbox. It again includes a cell stress panel, high throughput transcriptomics in multiple cell lines, and a multi-target in vitro pharmacology profiling panel. So our research is in progress. We're developing the data set now for about 100 chemicals. We are doing 4 cell lines worth of high throughput phenotypic profiling, or HTPP. This is essentially high content imaging of multiple subcellular organelles in the cell. And high throughput transcriptomics, where we're profiling the entire transcriptome that's available in those cell lines. We're pairing that with in vitro safety pharmacology for an enhanced view of both existing safety pharmacology panel and additional nuclear receptors of interest at the US EPA. We're using a developmental toxicity germline reporter assay, and we're using high-throughput toxicokinetic assays for informing in vitro to in vivo extrapolation of dose. So we can move from a micromolar in vitro concentration to something like a milligram per kilogram per day dose in vivo. This work increases scientific confidence in the application of this suite of bioactivity NAMs for informing chemical safety. And these data will be needed to evaluate the performance of this battery for future application. And it may also inform evolving frameworks for using multiple data streams. As you can see across the field, there's a lot of consensus building on how to do this. Finally, in research area 5, we've been collaborating with our colleagues in the New Chemicals Division to develop decision support tools for new chemicals. And I have a couple screenshots here, and I'll delve into some more details in the coming slides. But essentially what we're doing is we're using and demonstrating this tool, the cheminformatics modules, to primarily find analogs in a systematic way, make predictions for a target and its analogs, provide any publicly available hazard data that may be associated with that target and its analogs, and then any safety GHS information that's available publicly for any of those structures. And we have work underway to essentially continue to develop workflows for using this tool and look at any functionality that may be needed that we would need to develop. Additionally, we have some internally, well, I should mention from the last slide before I move on, the cheminformatics modules is publicly deployed, and you can find more information about it at the link at the bottom of the slide. We also have some currently internally deployed tools that we are moving to make external in the coming year, including this one shown to the right, abbreviated AMOS. It's Analytical Methods and Open Spectra. Here, for any chemical structure that you input into the tool, we can find documents, and those documents are analytical chemistry methods, available public chemical spectra data, and then this final part that I've highlighted, fact sheets. So we've also scoured the internet using a computational methodology to look for any fact sheets from other regulatory authorities, such as ECHA, the European Food Safety Authority, Health Canada, NICNAS, the OECD, etc. So we're really trying to create a document depot for target chemicals and analog chemicals so that it's easily at the fingertips of not only our external stakeholders, but our internal stakeholders as well. So this is a very early draft mock-up of how we might use the cheminformatics modules, and this is what we continue to work on in terms of draft workflows and SOPs for how this tool could be used. But in a nutshell, you can drop a chemical identifier into the search tab of this tool, and that could be a CAS registry number. We use DTX SIDs here if you're familiar with that identifier. You can use smiles, you can draw it. Any way you get it in, we can then identify analogs by similarity and tuning a set of rules around similarity and what you want in and out of your consideration. Then we can evaluate the utility of those analogs by sorting them by their available hazard information. So, what might make an analog useful is if there was a lot of information associated with it. We can then transfer a whole set of analogs, and I'll show you this on the next slide, to other modules in the cheminformatics modules. So once we identify, say, a whole cartload of analogs that we think may be relevant, we can actually look in batch mode at all of the hazard information that are available for all of those potential analogs. We can also look at the safety module where we present all of the THS data for any of those chemicals. We can transfer it to our alerts module where we implement alerts from the OECD QSAR toolbox as well as others. And we can search for any QSAR predictions, and we're working on increasing the number of QSARs that are deployed here, but it's essentially batch QSAR results for as many chemicals as you feed into that module. Then all of that information can be exported. So it can be provided for review and any further decision making. And then later this year, we hope to add that tool AMOS because we would like to make that document retrieval easy and seamless for our colleagues. So you can see here, this is a screenshot of the cheminformatics modules, and I've just put in bisphenol A. It's our favorite example chemical just because it has a lot of information and everyone knows it. So when I draw that, I can use a number of different parameters to find analogs. I can look for analogs based on similarity. I can set a similarity threshold. I can choose the kind of structure fingerprint. I can decide which elements should or should not be included. I can also do the same thing for substructure. I can say that I want a minimum type of toxicity information available, and then I can create a whole cart of potential analogs for further review and sending to these other modules, which are in the tab at the top. Here, I've transferred bisphenol A and all of its target analogs to the hazard comparison component of the cheminformatics modules, and it creates this table of summarized information, which you can click and go further and go back to the source. Bisphenol A is a very well-studied chemical, so you can see we have some amount of data at different levels of toxicity across a whole spectrum of human health and ecotoxicology effects. Some of its potential analogs are less well studied, and we have gaps in the data set. This is a way to rapidly develop a profile of potential analogs for your target, export these data and all the underlying information. It's reproducible and it's systematic. So what do we aim to deliver? Hopefully I've convinced you we aim to deliver a lot. It's very ambitious and it's ongoing. We have 5 different research areas. We hope to develop and apply chemical category and analog approaches for more rapid assessments, develop and deploy QSARs for a number of properties, develop an in vitro NAM battery proof of concept where we develop a NAM-based point of departure and fill potential hazard data gaps such as developmental toxicity, among other targets. And ultimately, we want to provide a connected system of ORD online tools developed in collaboration with OPPT for a more rapid, reproducible new chemical assessment. So what this looks like in a sort of final visualization is, NCCRP developing not only the tools to the left for data delivery. You may be familiar with our CompTox chemicals dashboard, and it really addresses some different kinds of inference level, like the specific chemical, and we do have some QSARs there. We have generalized read across in an online tool, which is really for chemical analog and chemical grouping. And then the cheminformatics modules provides a window into pretty much all of the inference levels. We already talked a bit about outputs from in vitro NAMs and how they can help us at these different levels of inference that are relevant for new chemicals. And all of this is built on ORD's database, DSS Tox, which is where we register and store chemical structure identities, and we're up to about 1.3 million there. So this project advances a number of goals across the EPA and ORD as well as in OPPT. Right now, we're building the foundation for continued improvement. It complements our EPA NAMS work plan, if you're familiar with that, where we've presented a forward-looking approach for what we intend to do in the development and characterization and confidence for NAMS. And then using NAMS to address EPA problems with resource efficiency. I think it's easy to see that, using some of these tools, we could really speed up certain processes and to further build our partnerships both in the agency and outside with other agencies. So with that, I'll give a very brief thank you. I wish I could recognize all of these people, but there are a number of people working not only on the research side to the left of the slide, but also those working in the Office of Pollution Prevention and Toxics, and really giving us a sense of what they need and how they need it so that we can come up with the right tools. So with that, thank you, and I will pass the mic to Rich Engler. Thank you for listening.
Yeah, Katie and Adam, I want to thank you both. It's fascinating to hear all the progress that's being made. It's very exciting. And I just want to touch, Adam, actually, you both touched on this. I wanted to come back and talk about why there's so much activity and why this is so important. So in the negotiations to develop the Frank-R. Lautenberg Chemical Safety for the 21st Century Act, there was recognition of the need for EPA to have data to make decisions about risk on chemicals, but there was also a clear need to strike a balance between that need for data and the use of animals, especially vertebrate animals. And so there was a fair amount of negotiation, and in fact, in the statute, there are specific requirements that EPA must undertake that strike that balance between the needed data and the use of vertebrates. So, number one, EPA is prior to requiring testing. So this is when EPA is going to order either through an order or through a rule, but prior to requiring testing, EPA has to consider what information it has. Does it have information that already answers the question or can be used to inform? Are there computational toxicology or bioinformatic methods that can inform EPA's decision, or are there other high throughput screening methods? So again, looking for ways to answer the question without new vertebrate animal testing. Clearly, Congress wants EPA to think about what's there, what's available. Does EPA really need to issue orders to orders or rules to require vertebrate animal testing? Next slide. The other is that EPA is required to, and I think what we heard today is it shows EPA is working diligently to encourage and facilitate new approach methodologies. So use of valid methods, to the point we heard earlier, fit for purpose methods that can be used to reduce or replace the use of vertebrates while providing information of equivalent or even better quality to support regulatory decisions. Adam mentioned that, in fact, some of the NAMs are even more human relevant than a mammalian model. So just because it's not testing in vivo testing in mice or rats doesn't mean that that it's not providing robust information to decision makers, whether it's EPA or another regulator or even the companies that are developing or deploying the chemicals. EPA is required to group into categories, and we heard about the old new chemicals categories, and it's a document that EPA developed over the decades in its review of new chemicals under TSCA, and I'm very encouraged to hear that it's seen as a resource going forward. I think there's a lot of useful information there, again, for companies, for EPA, for the public to understand what the hazards are of those categories, and to the extent that NAMs can help make those categories more robust or can provide other ways to test for the effects that are expected in those categories. I think there's a lot of exciting things happening there. And EPA's, I mean, this wasn't a heavy lift for EPA, but to encourage and facilitate formation of consortia. And we certainly manage a number of TSCA consortia, including testing consortia. But the idea that is, if EPA is going to order or require testing, whether through an order or a rule, that the orders or rules allow for the formation and allow for those that are required to do the testing to come together and do it in a consortia. So not everybody, not all, maybe 20 different manufacturers have to do the same test. They can come together and share the cost of those tests and minimize the number of times that test needs to be run, because the consortia is responding to that requirement. And EPA has a long history of encouraging consortia, but in what we're seeing now, or what we've seen since Lautenberg, when EPA has issued test orders, is that the orders are constructed in such a way that consortia are enabled, and the data flows within the central data exchange also allow for and enable the consortia to respond on behalf of the folks that are required to meet that requirement. So, right, there are these clear mandates. Congress's intent is very clear that EPA must continue to develop NAMs and continue to try to reduce the use of vertebrates when they are developing, or when EPA is either developing itself or requiring others to develop data to support decision making. It is unequivocal, and I think what we heard, especially from Katie, is that EPA is being very aggressive in this. The collaboration between OPPT, especially new chemicals, and ORD is very exciting, and I'm delighted to hear how much progress is being made. I haven't been as in the weeds with the collaboration. And so it's great to hear the update and how much progress is being made. I think it'll be great for OPPT. I think it'll be great for submitters if it's clear how data can be developed using NAMs instead of animal methods. There's a lot of opportunity here to help EPA make better decisions. And finally, just as my personal perspective is that there's just a tremendous amount of potential here to provide decision makers, again, whether it's an individual company or a regulator, with robust scientific information to evaluate risk of chemicals without over reliance on animal testing. And it's critical that EPA continue to support and that EPA be resourced to continue to support this. Not only is it required by the statute, but we are looking to get to a better place in terms of how we evaluate risk. So this effort within EPA really needs to continue, and I want to encourage people to think about how important that is, so that EPA is getting the support that it needs to continue to develop and deploy these NAMs in a scientifically robust way. So I'm going to stop there. I know we've got a number of questions in advance, and I saw that we had a couple questions come in, but let's go to the next slide. Lynn, do you want to switch to Q&A now?
Yes, thank you, Rich. Thank you, Adam. Thank you, Katie, for just the data richness of your presentations was just overwhelming. I am blown away at how much activity there is going on in an area, and just very pleased and proud that we are able, as a firm institutionally, to help amplify and communicate to a broader cohort out there of all of these amazing developments. Thank you for excellent presentations. The slides were very clear and very, very helpful. So thank you so much. We do have questions from our audience, and in the interest of equity, I'm just going to go through them in the order in which they were received, and we'll get through as many of them as we can. We have about 20 minutes, so, and I'm going to throw these out to all of you to comment as appropriate. And if you don't wish to, just say pass, or I'll move on. So the first one is, what can be done to improve the confidence of regulators when presenting NAMs-based test data for chemical safety? Regulatory agencies, while expressing a desire to increase the available NAMs for chemical safety, have been, in some occasions, as you all know, somewhat hesitant to adopt them. So, Rich, do you want to start out, or?
I know. Actually, I want to defer to Adam and Katie
Okay.
on this, because they're really doing that. Right. That's part and parcel of what they're doing.
Right.
It clearly, there's a mandate that EPA use fit for purpose validated NAMs, but they're the ones that are doing the work. So I'm going to defer to them.
Yeah, and I would really point to the ICFAM report that I referenced in one of my slides that came out last year. I think that's a great resource, great place to start there. Improving the confidence of regulators, I think, really comes down to those key concepts, where you look at the biological relevance, data integrity, technical characterization, information transparency, context to use, all those different pieces together. And that ICFAM report does outline that very well. So I would suggest to reference that, and that's a great place to start.
Thank you, Adam. Katie, you want to add anything?
Yeah, I agree with Adam. The ICFAM report has all the elements I'm thinking of. I'll underscore a couple that we're working very actively on around NCCRP. One is peer review and making the data public. So Adam just mentioned that, but having all of the data and methods be public and transparent and peer reviewed, I think, is one of the key elements, several of the key elements in the ICFAM report. Another is working in consortium like we do in the Office of Research and Development, that APCRA consortium I mentioned, where we're really working with the global community of scientists in regulatory agencies at Health Canada, European Chemicals Agency, European Food Safety Authority, NICNAS, A-STAR, really bringing people to the table, and then publishing case studies that evaluate, how well do NAMs work. So I think that's an important piece of the puzzle. And then the last one is, working through the OECD, right?
Mhm.
And so we have efforts at the OECD to try to push forward certain types of NAMs, but also now collections of NAMs for things like systemic toxicity.
Yeah, and the one thing I would add is something that Adam said at the very beginning, which is a NAM may be in fact more human relevant than a rodent model. So, to the extent that that these NAMs are well constructed and well founded, they may be even better, not just a replacement for, but superior to rodent testing.
All right, thank you. Next question. What level of information or evidence would be required to get NAM results accepted during an LVE, a low volume exemption, exemption request under TSCA Section 5, or pre-manufacturer notification procedure at US EPA? Regulatory acceptance of non-animal testing is often not yet obvious.
Well, I'm happy to go first on this because I'm
Sure.
not sure. I'm not sure Katie wants to step into OPPT's role. So there are a number of pre-vetted NAMs that OPPT has that they've published lists, and we found that they generally do accept NAMs that on their pre-vetted list. So that's sort of the easy answer, is these are the ones that OPPT has already said are acceptable. If you're starting with something new, you're going to have to convince them, and it's going to be a matter of showing that it's, the graph, that Adam's graph that he had early on, that, is it fit for purpose? What's the basis for concluding that it's predictive? That's going to be a heavier lift, but it doesn't mean that you can't do it. It's just, to the extent that it takes a while to develop the science to show that something is fit for purpose and acceptable, that's going to slow down a PMN or an LVE review. But it's not that it can't happen. It's just going to be hard to do in a timely way.
Thank you, Rich. Any other comments? All right, I'll move on. Next question. What is your perspective of Daphnia testing for aquatic toxicity and the pressures around non-animal testing methods?
I'm not sure. So the requirement in the statute is to reduce vertebrate testing, and Daphne are certainly not vertebrates, but they are animals. So there's a little tension there. But at least from a statutory standpoint, testing with Daphnia or other invertebrates is not something that EPA is required to develop NAMs to do. It doesn't mean that there aren't NAMs. I mean, certainly EPA uses QSAR methods to predict aquatic toxicity, but they are not. There's not the same statutory pressure for EPA to develop NAMs to reduce invertebrate testing.
So it sounds like it's within the spirit, but not necessarily the scope.
Sure. Yeah.
I'll add onto that, just, for the sake of time, I didn't show the methods that we're working on for rapid ecological assessment, but some of the methods we're working on are essentially modifications of standard protocols like those, including Daphnia, right? And doing that in a 96-well plate, and then using RNA-seq technology to come up with a transcriptomics-based point of departure. So essentially making more rapid and miniature some of these models for Daphnia, and then 3 other taxa is part of our approach. So not really commenting on moving away from it so much as it's still very much part of our research of how to provide a rapid, eco-relevant point of departure, particularly for chemicals that might not yet fit the domain of applicability for a model like EcoSAR.
Great. Thanks, Katie. Next question. How do you determine if an assay is appropriate for a class of reactive chemicals, i.e., if you are testing the actual chemical or a hydrolysis product?
Well, this is a problem whether you're doing a NAM or an in vivo testing, right? It's always a challenge to know what what should the test substance be, and part of that is the study design, making sure that you are you know, and part of it depends on what are you evaluating. Are you evaluating, you know, what a worker might be exposed to in a workplace where it's probably the intact substance prior to hydrolysis, or are you looking at potential risks from, say, drinking water where it's already hydrolyzed and someone's exposed to the hydrolysis? So it's both, I think, are potentially relevant in a risk evaluation. But I'll let Adam and Katie talk about which one you would test in a NAM.
Yeah, I think going back to kind of that donut chart that I presented about the considerations for developing a NAM, you know, that comes down kind of into step one of the chemical properties of what you're looking at. And you can do some upfront work, like Rich just mentioned, as far as doing some stability testing, some reactivity testing, and then really getting to know your chemical, physical chemical properties. And, you know, things like, is it bioactivated? Are you looking for that metabolite? Is that actually what's causing the toxicity? So I think the more you can do upfront to really understand the chemical and how it works in the biological system, then you can work to develop the NAM and try to find the right fit for purpose with that. Yeah, there it is.
There it is. Thanks, Emily. Katie, anything to add?
I think in the interest of time, we can move on, except that this is an area of active research for us where we're looking at, you know, what are even the physical chemical properties that are screenable in different in vitro settings. And, you know, if you're interested in inhalation versus oral exposure, depending on the media, right, you might choose different NAMs. So this is part of what we're doing in our proof of concept work, is focusing first in an area of applicability for aqueous-based screening. But of course, you're going to need more NAMs to cover things like inhalation, where you might be looking at aerosols or different physical chemical properties that maybe are not even bioavailable in a largely aqueous system.
Yeah, just difficult to test is difficult to test in any assay, right? So it's just, they may be a little bit different in a NAM, but it's still a challenge that you have to address when you're doing testing.
All right, I will move on. Thank you all. Next question, is ICFAM or NICEDM working on validation and acceptance of GARD air?
So I can't speak for ICFAM or NICEDM directly, but so GARD air is a respiratory sensitization model. And the OECD has said that they are working on a review paper on that. And I believe the last I've heard is that target for that that review is to come out next year in 2026. So there is work being done. There was another area I found there where NIEHS and collaborators are testing about 100 substances nominated by U.S. federal agencies using GARD air, and results for those tested chemicals thus far indicate that hazard classification using GARD air shows some promise. So I believe it is in the works. Whether it's official validation, I can't quite speak to that, but there is work being done on it.
Oh, that's very helpful. Thank you, Adam. Shall I move on? All right, next question. How is EPA's current definition of NAMs, which includes new animal tests like the ETAP, related to TSCA's mandate to, quote, promote the development and timely incorporation of new scientifically valid test methods and strategies that are not based on vertebrate animals, end quote? And a quick follow-up, will the TSCA list of alternative methods and strategies include animal tests like the ETAP?
So maybe I'll start and others can fill in. So the ETAP, if you're not familiar with it, is the EPA transcriptomic assessment product. This is based on a 5-day rodent study and then transcriptomics in 10 different tissues. It was reviewed and accepted for the development of point of departure. And there's a couple of different answers to this question. One is that I don't think anyone anticipates an all or none approach to animals. I think it's about using animals when it's appropriate. The other is that this ETAP is really intended for extremely data poor chemicals. So these are chemicals that wouldn't have any other, 90-day study information. And so it's really a bridge because right now there's no accepted and FACA reviewed point of departure methodology using in vitro technologies. So this provides a bridge to that in a short-term animal model. And the other is that, ETAP is something that can be used by a variety of constituencies, states, tribes, across the agency for data poor chemicals. So I think the use is even broader than just the TSCA space and could be used by different constituencies of the US EPA.
Mm-hmm.
Yeah, and it's certainly a consideration if it's a substantially fewer number of animals that are required to get a similar data set, then I think that would meet the right. Not being a lawyer, but I think we could argue that that meets the requirement that EPA is reducing animal testing, even though it with a NAM, the ETAP NAM, even though it hasn't eliminated that animal testing.
Yeah, that's correct.
Move on. Thank you very much, Katie, for that, and Rich. Not discussed as much as the economic impact of NAMs on contract research organizations, otherwise known as CROs. Are there data out there, or maybe you can speculate on any impacts as CROs transitions from traditional animal testing to non-animal approach challenges? What might these challenges be, and are there any better summaries of how significant they may be or how CROs are transitioning? I think the premise of the question is that CROs seem to be based on or grounded in more traditional animal testing, and moving into a non-animal testing framework may be challenging or not. What are your views?
Yeah, I mean, there's no question that CROs are going to have to develop the capacity to do the NAMs. Right? They've got their traditional business, although there's some EROs that do focus on NAMs. But to the extent that NAMs become more widely available, more widely used, more widely relied upon, I think we will see the transition for the CROs to move away from vertebrate testing to NAMs. It's going to take a transition. I mean, the science of the NAMs has to come first, and then the reliance of the NAMs, and then then submitters or agencies are going to start placing NAM testing. And so the CRO capacity to do that NAM testing, I just expect a lag.
Anyone else wish to comment?
I'd say too that there are some CROs that are very specialized in NAMs testing already. So I think we've seen, some of that transition already happen.
Great. I think, Adam, this question really goes to you. You spoke about validation, if that's even the right term. Not all NAMs are fully validated at the international level before they may be considered fit for purpose or valid for assessing environmental and human health hazards. Does this impede regulatory acceptance? And if so, what can be done to lessen this barrier? I think we've all kind of talked a little bit about that, but maybe you can give some short pointers to be directly responsive to that question.
Yeah, I think again, I'll harp on going back to that ICFAM report and what goes into the development of a successful NAM if it isn't quote unquote validated or accepted international method. And then the other thing I would just I'd really say work on collaboration. And if it's something that you would like to submit to the EPA, do your best to keep open lines of communication there. Let them know what you're working on. The sooner the better that you can inform them, get that feedback, and make sure you're on the right path. And hopefully that keeps you moving in the right direction.
Yeah, answer the right. You need to know what their questions are so you can answer them as part of the development. Yeah.
Great. Thank you, gentlemen. Next question. Some suggest that it is important to conceptualize NAMs to manage expectations. The thought is NAMs are not intended to replace, in all cases, animal testing, but rather incrementally to improve ways to assess human hazard and risk. Does this make it harder to quantify the value of NAMs? particularly to those who are most comfortable with traditional established test methods?
Maybe
I
I'll,
think the answer is yes.
maybe I'll start off. This is a very interesting question and very thoughtful. So one place to start is a paper that Patience Brown, Rusty Thomas, and I published in 2024, which really outlines what is the existing framework for toxicity testing and how does it parallel a NAM-based testing framework.
Oh, it's
And
perfect.
the conclusion is really that we're trying to do the same thing. You have NAMs that you develop because you're trying to develop a protective point of departure. You're not necessarily trying to predict a specific effect in humans. In fact, it seems doubtful that that specific effect, might occur, like a decrease in body weight, for example, is, common finding in an animal study, commonly underpinning point of departure, right? But it's not that we actually are trying to predict that effect in humans, right? So it's protective point of departure. And there's a toolbox of NAMs for that. And then assisting that is a toolbox of NAMs for prediction of specific hazards, right? And so all of these are at different states of readiness. And in the meantime, we have to continue, in a toxicology framework, right? So you see NAMs at some state of readiness contributing to weight of evidence. You see NAMs at some, readiness contributing to development of point of departure, right? But if you think about the current framework, we have traditional toxicity methods that are really aimed at protective point of departure, and then you have specific studies for specific types of hazard, and it's really the same for NAMs, and we're just trying to build that toolbox. So that may not directly answer the question, but
No, it's hugely
that's
helpful.
how I hear it, because I think we are really trying to build essentially the same framework, the same way that animals are used. That's how NAMs eventually will be used.
Gentlemen, anything to add?
No, yeah, that's a great summary.
All right, well, this will probably be our last question then. Excuse me. NAMs for local toxicity have been validated by correlating the results with animal test results and applying uncertainty factors to make a safety determination. For more complex endpoints, however, like systemic toxicity, we seem to be a long way off from making safety determinations that do not involve animal data. Two questions. First, is this a correct summary? And second, will AI be of any use here?
Well, I guess it's unclear to me when you say not relying on animal testing. There's certainly, as part of the review and validation, there's some comparison between the NAM and existing in vivo data. So to that extent, yes, the existing in vivo data in vertebrates is informing the validity of the NAM. But I'm not sure that we need, once we've figured out that the NAM works, I'm not sure that we necessarily need the animals going forward. So I'm not quite sure what the questioner was getting at there.
Okay, fair enough. What about AI, Rich?
AI, look, AI is a tool, and
Right.
and whether the tool is well constructed is how do you build the AI?
Right.
A generic AI that's built off of scraping the internet is not going to be good for this. But if you develop an AI using, all the extraordinary amount of data that Katie talked about, I think that could be a tool that, that's used to help inform NAMs, validate NAMs. Certainly, doing in silico calculations, AI could be useful for that. It's all about how do you design the tool.
Katie, Adam, anything to add?
I'd love to jump. Oh, sorry. Go ahead, Adam.
No, that's okay. I was just going to say, touching on specifically the systemic toxicity point of it, even with Katie's presentation, I think it shows how much work is being done, even with it is very complex, and more complex than, something like simply assessing skin irritation. But there is a lot of work being done and screening work being done, and there are in silico tools, there are modeling tools that can look at things like that. So yeah, there's I would say that there has been significant progress in assessing systemic toxicity using NAMs, and there continues to be.
Great. Thanks, Adam. Katie, we're going to say something?
I mean, Adam said it really well. I think a lot of progress has been made. You have to think about it, I think, from the traditional lens, right? So what is a traditional toxicity study that would be considered adequate for assessing systemic toxicity? A 90-day subchronic study, oral exposure. What are the NAMs I need in a battery to be able to provide an alternative to that? Might be different than the NAMs that provide an alternative to an inhalation study for systemic toxicity, right? So it's block by block building that. But I think we are getting closer, particularly for oral systemic toxicity and alternatives to a 90-day.
Great. Thanks, Katie. And with that, I'm afraid we need to conclude our webinar today. I want to thank our fabulous presenters, Adam Bettmann, Katie Paul Friedman, and Dr. Rich Engler, for their really extraordinary overviews. I know I have learned an awful lot, and we are so appreciative of your willingness to share your thoughts, and the updates that you provided here are just incredible. Thank you for all of your work and contribution. Katie, thanks for all that you do. Give our best to all your colleagues at EPA. We're with you and really are very appreciative of you being here today. Adam, same. I hope everybody enjoyed this as much as I did. And just a quick reminder, if you like what you heard today, we have a bunch more webinars scheduled in our 2025 webinars series. They're all complimentary, and they are all done as professionally and as brilliantly as this one. So thanks all very much. Really appreciate it. Have a great rest of the day.